an assumption of Hardy-Weinberg that populations are isolated, but a population is seldom an island, but rather exists along a mainland where other populations can interact with it. In this lecture, we're talking about migration and the movement of alleles between populations as a violation of Hardy-Weinberg equilibrium. I'd like you to be able to define it and to contrast it with selection. Also be able to define local adaptation and give at least one example, perhaps something to do with snakes. So what is migration? Migration is a movement of alleles and the organisms that carry them between populations. Let us say, for example, that we have an island existing perhaps off the coast of Africa. And this island off the coast of Africa occasionally gets certain individuals coming from the mainland to it. Perhaps these are individuals of some sort of wild mustard species. So this wild mustard species occasionally has some seeds traveling on the feet of birds over to this little island off the coast of Africa. Over time, there will be a population of wild mustards on this little island, and it will be exchanging alleles with the mainland. Now, as we might notice, that we have an unequal exchange from the island to the mainland. Birds coming from the continent, where there are plenty of wild mustards in large populations, are going to be bringing a greater number of alleles and a greater number of individuals than birds coming from the island back to the mainland will bring. Thus, we can see quite quickly that migration really is a deal where a larger population can swamp out certain adaptations in a smaller population. We also might think, why might one population have a different set of alleles than another? So one of the simplest reasons for a population of a different number of alleles is simply genetic drift. This might be founder effect. So perhaps the first mustards that came to this little island off of the continent had white flowers, an odd thing for a mustard to have, really. We think of yellow. But what happens is these white flowered mustards just so happened to be, it's a population that has that allele more frequent because some of the founders had a higher frequency, a misrepresentative frequency of that white flowered allele. So perhaps that island population has white flowers on it. And that's just a chance founder effect deal. Perhaps there was genetic drift, where there was just a drift towards the white flower allele over time on the island just by chance alone. So these two things might be reasons for the island having a different type of set of alleles than the continent. And migration will alter allele frequencies. So we're going to think this in terms of migration is going to bring new alleles and more allele combinations. Different frequencies. So let's say you have this population of mice here. And this population of mice has the initial allele frequencies of 1.0 in this A1 allele. So the A1 allele is 100% of the population. 100% of the genotypes are A1. 100% of the juveniles are going to be A1. But if 200 of these A2 individuals, they're homozygous for A2, are going to move in from the mainland, we're going to end up with 800 A1A1s and 200 A2A2s, and that's going to make a new allele frequency. Now that's kind of an extreme example where a new allele and a new allele combination moves in from the mainland to the island. What we can also see is that allele frequency that if the mainland has 80-20 and the island has 100-0, that any movement from the mainland to the island runs the risk of changing that frequency because it's introducing a new allele. Migration also tends to homogenize things. Alright, so what I want you to think of is a young population, an intermediate population, and an old population of something called red bladder campion. It's a type of flower, and you can see it on figure 7.3 in your book. Some researchers were looking at populations of red bladder campion on this archipelago here near Sweden. What happens is young populations are founded when a whole bunch of genetic diversity comes along in a recently disturbed field. These are organisms that are going to be kind of invading newly disturbed habitats. Intermediate populations are going to be those that are pollinated by other populations because as they get intermediate, they're going to have more pollination kind of coming in and whatnot. And old populations are dying out because there's this disease that comes by pollinators, there's competition because these are early founding species. And what happens over time is you see genetic diversity. So you see the amount of genetic variance on these young populations between populations. genetic variance between populations is very high. Genetic variation between populations is very high because of things like founder effect, where some of these populations were founded and they rapidly expand. So they're going to be mainly one genotype in population A and mainly one genotype in population B. Intermediate populations have less genetic variance, seen here by the error bars, these smaller error bars, and that's because they're pollinating and working with other populations. So your genetic variance is relatively low in intermediate populations because of the migration of alleles between populations. Whereas old populations, you just have some survive and some don't, and the random chance of who survives and who doesn't is going to make for more allele variation. So the variation is lowest in populations that have migration of alleles in and out of them, which shows that alleles are going to be moving between populations and end up homogenizing it into what we call a metapopulation. A metapopulation is a whole bunch of different small populations that are capable of interbreeding with one another and recolonizing. So take that archipelago that I can't pronounce once again, and what you're going to see is some of those islands are going to have a population of red bladder campion and some of them aren't. The ones that aren't can be colonized by the ones that have it. The ones that have a population that is relatively low in genetic diversity, the population can increase in genetic diversity if genetics move from one island to the other. So this metapopulation functions like a single population made up of smaller populations. So that's really what we're seeing here in migration, is the homogenization of the gene pool through migration of alleles between subpopulations of a metapopulation. Whew. Okay. Selection, on the other hand, is going to specialize. So selection is going to cause what are called local adaptations. So here we have a group of snakes that are from Lake Erie. So what happens is these water snakes generally have this banded pattern, making them a little more difficult for predators to see. However, this banded pattern is not adaptive in certain environments, resulting in different selective pressures, resulting in different populations. Natural selection decreases genetic diversity by weeding out bad alleles. So alleles that are not adapted to the local conditions are generally have a lower fitness and thus will be weeded out of the population. So natural selection specializes a population by getting rid of bad alleles, maladapted alleles. Natural selection decreases genetic diversity. But migration is also often at odds with local adaptations. So we look at these, these islands in between Ontario and Ohio. And these are the, these islands here have populations of snakes. Now on these islands, we're going to have a lower amount of genetic diversity, and we're also going to have certain adaptations. So for these ABC genotypes, as it were. So what percentage of the population is made up of ABC genotypes versus D genotypes? Well, on the Ontario mainland, it is strictly the D genotype, because that is the genotype that is most adapted to the Ontario mainland. On the peninsular mainland, we're talking Ohio, you're going to see A, B, and C genotypes at very low frequencies, but the D genotype dominating, because again, the D genotype is the most adapted, but A, B, and C are not so maladapted that they cannot exist. However, on Kelly's Island, Bass Complex Island, and the Middle and Pelee Islands, you actually end up seeing the A, B, and C genotypes dominating because those genotypes are more adapted to the local environment. It's these different banding patterns or being smooth-skinned and not having any banding patterns are the different genotypes. So what type of snake it looks like depends on what rocks it's basking on, which is going to give rise to these A, B, and C genotypes being more advantageous on the islands, and the D genotype being more advantageous on the mainland. However, migration is going to occur here. So those well-adapted snakes, the A, B, and C genotypes, are going to see migration from Ontario and Ohio that are going to bring the D genotype, which is maladapted. So the introduction of those new alleles is going to be at odds with the elimination of non-adapted phenotypes. These new D alleles just keep getting dumped on the doorway of these islands, and they keep getting weeded out. If there were, say, some sort of water wall that was preventing migration of the maladapted phenotypes, or genotypes, then you wouldn't see the D alleles at all on Kelly's Island, the Basque complex, and especially not on Middle and Paley Islands. You wouldn't see those genotypes because natural selection would weed them out. So migration and natural selection are often at odds because migration is going to homogenize populations and natural selection is going to result in local adaptations by the specializations through natural selection. So there we see how migration and natural selection both violate Hardy-Weinberg equilibrium, but often in very different ways.